That was so bad. That delivery was so garbage. Yo, what's good, fellas? We're talking about DNA today. Basically, it's a self-replicating material which is present in nearly all living organisms. It is the carrier of genetic information. The two strands of a double helix DNA strand are made up of phosphate and sugar. In between the strands are the four bases: adenine, thymine, guanine, and cytosine. Hydrogen bonds hold those bases together. DNA is also anti-parallel and has different ends: the 5' end and the 3' end. The 5' end is made up of phosphate and the 3' end is made up of sugar. The first step of DNA replication is to separate the two strands. The helicase, an enzyme that uses ATP hydrolysis energy, or GTP, to catalyze the opening of nucleic acid, unzips the double helix to allow replication and transcription of our DNA. Once the replication fork detaches the two strands, we are left with one primase. The primase is an enzyme that creates short RNA sequences called primers. A primer is a molecule whose presence is required for information of another molecule. What does that even mean? From the primer, the DNA polymerase starts to make the new DNA strand. It is only able to add bases in one direction, from the 5' to the 3' end, which starts with a phosphate and ends with a sugar. The leading strand that the DNA polymerase has now created is then made continuously and constantly, adding more bases. The lagging strand that is not being used to create more DNA cannot create continuous bases because it is going in the opposite direction. Instead, this strand is able to replicate itself in small chunks that are called Okazaki fragments, which are basically just short sequences of DNA nucleotides. Start with an RNA primer. Another DNA polymerase begins to replicate this strand. It has a short row of DNA in the 5' to 3' direction. Another primer is added further down the line and continues this process with another DNA polymerase. This process continues down the strand. Once the new DNA has been created, the exonuclease removes the RNA primers from both strands of DNA. Another DNA polymerase fills the gap in with DNA strands. The ligase, an enzyme that can catalyze the joining of two large molecules by forming a chemical bond, seals the DNA in both strands to form a double strand. DNA will always be made up with one old strand, and the replication process is now done until the old strand needs to be replaced. Thanks for listening.